Okay, so this is going to be the Wednesday, December 11th study session, focusing on the semester 1 exam review. And we're going to start here at the beginning, sort of, and see how far we go. Your questions, your interactions, your responses is going to kind of shape how we kind of drive this. Does that make sense? I don't have like a PowerPoint I'm going to go through with you guys. We're just going to talk about it and do some practices as we go through. Cool with that?
Yeah.
Okay, so let's start at the very beginning of the year. In the very beginning of the year, we had those sponge eggs. You guys had to evaluate the claim evidence reasoning. Do you remember that part? Yes, you had to do little animals, your babies. One of the reasons we did that is so we could review experimental design. You have had experience setting up experiments before, but it was sort of to kind of review and kind of hone in on how to do it. And then we had lots of experience when we did placing science. So when you're doing this, you need to be able to write out a problem. A problem is like a purpose, what you want to investigate. Hypothesis and procedure. The biggest issue we still have, kind of, even with planning science, I wrote it on some people's, is that the hypothesis, don't think of it as an educated guess. That's the prediction more so, even though a lot of people say educated guess. But it is a testable answer to a question. So if I ask you the question, or the purpose of my experiment is to see what helps a plant grow. What will make a plant grow faster? That's the purpose of my experiment. What would a hypothesis be for that? Give me a hypothesis that you think is an answer to the question that we can design an experiment to test it. Jacob.
What's the question again?
What can make a plant grow faster?
Fertilizer A.
Okay, so a fertilizer can make it grow faster, and we can be specific. A certain brand of fertilizer can make it grow faster. Okay, that would be your hypothesis. It answers it. We can design a test on it. Okay, the prediction would be based on your experiment. You can make your hypothesis first. You make your hypothesis first, then you design an experiment to test it. You want to test the fertilizer to see if it answers the question. You think it's going to make it grow. Okay, you have background knowledge that it contains nitrogen and phosphorus, things that plants need now that we've learned about that. So it's based on research. Now my prediction, though, I've decided to test different plants. Plant with fertilizer A, like Jacob said. A plant with no fertilizer. I'm going to test a plant with fertilizer B. My prediction is which one of those will make it grow fastest. If I add fertilizer A, it will grow faster. I'm being specific to how, or if I add it once per day, it will grow faster. It's specific to my procedure. Does that kind of help clarify it?
Yeah.
So ask yourself these questions if you're not sure. Yeah.
So the hypothesis would be that something would impact it.
So like fertilizer impacts plant growth. Okay? Fertilizer affects the rate of the plant growth. If you said just fertilizer in general will make plants grow faster because it's full of nitrogen, that would be fine. Okay? That's kind of borderline prediction, but it's still general statement. It's very general. The prediction gets more into the nitty-gritty. Yeah.
So in this case, the prediction would be like this amount of soil.
Yeah. Yeah.
Whereas the hypothesis would be soil effects.
Yes.
Yeah.
You're just more general, not based on the experiment. Variables, most of you guys are doing pretty well at variables. Identifying the independent and the dependent. Independent is the one that you change up, you manipulate. Dependent is the one you measure. One thing that happens is people get control group and experimental group mixed up. The experimental groups are the ones that you apply whatever it is you're testing to. The ones you put the fertilizer on. The ones the experimental group would be the ones that you treat. you add something to. You put under certain conditions that you're interested in. And so sometimes I've noticed people still get the experimental and control group mixed up. But you will want to be able to test that. So I pulled up just, I'm not going to do the extensive ones, the ones with lots of reading. We can always do this at the end. But let's see if we can practice. Can you guys read this from where you're at?
Yeah.
Okay. And I hope I did that right. Oh, crud. I tried to plus it, and I might have just deleted it. Let's go Control R again. Oh, no, I just made it down further. Okay. So a cow is given a growth hormone and compared to another cow that was not given a growth hormone. Both cows were weighed at 2 years. What is your independent variable in this situation? Jacob?
The growth hormone.
The growth hormone. That's what you changed up, adding it versus not. Dependent variable, Brady? What is it?
The weight after 2 years.
The weight after 2 years. Okay? What about number 2? Mosquito repellent is sprayed on one arm and the other arm is not sprayed. The number of mosquito bites is counted after 2 hours. What would be the independent variable here? Ava? The repellent. And what is going to be, Christian?
The number of mosquito bites.
Okay.
Or mosquito.
Yes, the number of mosquito bites. Now, here's the deal. I'm going to write that down here. 2 things. Number 1, in this case, what is the control group? Which arm is your control group? Do you think, Michael?
The arm without mosquito repellent.
Yes. The arm not sprayed is your control. The one that you're actually treating is this one that gets sprayed. That would be your experimental group. You always have one control group, but you can have lots of experimental groups. So you can have an arm that had 5 milliliters of spray, an arm that had 10 milliliters of spray, an arm that had 15 milliliters of spray. That would be 3 different experimental groups. That sort of makes sense? One control, lots of experimentals. Possible. Now, my one thing here is the one issue with variables I do notice, and it happens a lot, is I will get something like bites as my answer for your dependent variable. What is wrong? Why would I mark that wrong? I'm going to give other people a chance to over on that side. Why would I mark, if someone just wrote the word bites, why would I mark that wrong? How is that different than the last answer? What is it?
Not specific.
It's not specific enough. If you just write the word bites, I don't know if you're measuring the number. I don't know if you're measuring how big they are. I don't know if you're measuring what color the bite turned. You have to write down, you have to be specific. A lot, and that has been consistent throughout the different times you guys have done like planting science and photosynthesis labs and the beginning labs. People will just be general here. So you need to be specific. What unit will you be measuring it? And think about that. Numbers, centimeters. Okay. Questions on that? Yeah.
Okay, what's the difference between an independent and dependent? I think we already said it, but I don't
Yep. Independent means it's you're changing it. You have control over it. It's being manipulated. It doesn't depend on anything else. Like you can control it. Okay, you're making the change as the experimenter. The dependent one is going to be changed because of whatever the independent was. It's what's changing. It's what's responding. They often call it a responding variable. It's what's responding to that change in the independent. I always use the word measure because oftentimes you can ask yourself, well, what is being measured in this? Because a lot of times you're measuring something in science experiments. Okay? The next part is on this part right here, talks about graphing. So while we're talking about variables, you guys have been doing, 7th hour today started on the population growth activity, and you had a graph in that activity. And when I was walking around, I was very, very happy because you guys tend to have a good grasp on doing your graphs, and more so than don't tell last year's bio groups, but even more so as a whole than last year's bio groups. But don't forget to check to make sure that you've covered your tail. Okay, or you covered your tails. I know, funny. When you do this, meaning your title, your axis, your intervals, your labels, and your scale. And you should have notes in your notebook about that. Scale is that you want to make sure that you're graphing over the whole thing. You don't want to keep it all scrunched down. If you do this using sheets or Excel, it pretty much does that part for you. But if you're doing it by hand, you want to make sure that you take up the whole graph. And you do that because of what you set your intervals at. Well, the scale is actually your minimum to maximum, and then your intervals is what you divide the lines up to. The easiest way, if you do struggle with scale, the easiest way to do it is to take the range, okay? Look at your minimum and maximum, set it at different ends, and then to figure out the intervals, count the number of squares, and divide your range by your squares. Christian.
What was L?
I haven't hit that one yet. I skipped it. I and S kind of go together, intervals and scales, so I purposely skipped it. A is axis, meaning you know that that's X and Y. And does your independent variable go on your X or your Y? The one you change up. Say it louder. X, okay? So knowing that your independent's here and what you measure or your dependent is over here. Okay? I'd say like most people get that. Sometimes you'll see it backwards, a flip-flop. But
Malaysia Vincent, please come to the office. Malaysia
Yes.
Vincent.
Okay, so if your graph, like, let's say if you're looking at your graph, if there were a space between 0 and when you first came in, if it were a small or a large space, would you graph it differently because of scale?
are you talking about like today we started a population and it began in like 1965. So we instead of starting at 0, we started at 1965. Is that what you're asking? Okay. So in math and a lot of times in science, you use 0, 0 as your starting point. However, if you do have a big gap, it is okay to not use 0 as your starting point. It is okay to use your starting point. And when I say starting point, that's where the X and Y meet up. You could start at 1965 and then still start at 0 for the population going up. That's okay. What I would suggest not to do though is, because then you get into a whole other can of beans, is not to go 0 and then 1965 and then go on from there. Start the 1965 at the axis. If for some reason, sometimes you will see this big gap, and they always have to, you guys have seen the squiggly lines? Have you seen that before? So, I mean, it's possible, but try not to, okay? I don't think I'll give you anything that will require that, yeah.
But what if you're measuring something in between a few minutes and it's just like flat lining at 0 and then it shoots up? Could you like shorten it down, or would you have to write all of that?
Well, you'd still have it. It would be like this.
Okay.
And that's better. That's what you want to do. Yep. And you'll see that a lot with population sizes. Questions on that? Okay. Now, I will tell you this. If you're interested, you can let me know before we leave here today. These are extensive ones. Do you remember at the beginning of the year we had I gave you scenarios and you had to identify the variables? I have other ones here. Now on the test, your scenario would not be quite this long, okay? It would be more like the ones we had at the beginning of the year, but if you're interested in some extra practice, what I can do is go ahead and print these off, and I can have these for you tomorrow. Is there anyone interested in having me print these off of that? Do you think other people would be interested? Okay, so what I'm going to do, just so I can remember here, and Melissa, I'll get your question in a moment, is I'm actually going to hit print now, so that way I'll remember to get it printed off for you guys. What was your question? Oh, I didn't. You're right. Or T. Darn it. Sorry, guys.
Title.
T was title. So before we can do the T though, we have to do the L. What does L stand for?
Labels.
Labels. And what do you have to have with your label, or else you might get docked, or you will get docked for sure? What else? If I said like distance, what else would I have to include? Units, units, units. Make sure you have those units in parentheses. Okay? To do the title then, how do I write the title? What do I need to make sure to include in the title? I don't like frou-frou in science, okay? I want to get straight to the point and let someone know what is going on, what this graph is about. So what do I need to include, Jacob?
Part of the question.
Not necessarily part of the question.
Both labels and their units.
Both labels. Units, sometimes depending on the question, yes. Okay? And so the cheat way I told you guys at the beginning of the year to do it is something along the lines of your Y variable compared to X variable. Distance compared to time. Growth compared to amount of fertilizer. You can tweak that part up. You can use words like versus, compares, changes, things like that. But you want to make sure that you get both these labels in it. You don't want it long. You do want it to be descriptive. If you have multiple groups, like say you're testing the fertilizer on different plants, you would do growth versus fertilizer of different plant species. So you'd have to include that you're doing different groups. That would be a good thing. For this, at this point, I wouldn't deduct you for not having the multiple groups, as long as you have what? If you have multiple groups and you have multiple lines, what do you need to include? Key. So make sure you do have a key if you have multiple lines. Unless told, well, we did talk about different ways to do graphs. We had like the histogram and the bar graphs. We had the line graphs. Line graphs are numbers. Most of the time we'll end up doing line graphs. We do a lot of measuring. Bar graphs are in times of discrete qualitative items, like the color of something. If you're comparing the color, amount of fertilizer of blue roses versus red roses. Okay, we might have blue and red. Well, no, that would still be a line graph because it's fertilizer in time. But if you're looking at like the number of something, okay, the number of flowers on the red rose and the number of flowers on the pink rose bush, that would be an example of a bar graph. Yeah.
Well, and then bar graphs are spaced apart, right?
Bar graphs are spaced apart. Histograms aren't. Don't stress about that.
So we should write that down.
Nope. We talked about it at the beginning of the year. It's good to know. You'll cover that in more detail as you go further. Histogram is like bar graphs that are squished together. There's no space. And the reason that they're squished together is, what do you remember about a histogram, Christian?
Over time.
It's not necessarily over time.
They cover averages.
It's not necessarily averages, but it's over things that are continuous, like height, the number of people at different heights, because we have 5 foot, 5'1", 5'2", 5'3". It's like continuous amounts. But that gets a little nitpicky. I won't be asking you to make a histogram versus a bar graph. Does that make sense?
Will we have any bar graphs on the?
I don't know. I'm being specifically vague. I would study line graphs over bar graphs. That would be all I say to you. I know. Yeah.
Is our test on the computer or by?
More than likely, my gut is I prefer it by paper for this. And what I might do is, if you took, I think it was the photosynthesis review, like the second photosynthesis quiz on paper, I had those like little Scantron sheets for the multiple choice section. I might have that.
Will you scan it for right in front of us?
On my phone. I won't scan it right in front of you, but I'll scan it that day. I'll do it just like I did with the review quiz. As soon as I can, I will. Okay? I don't want to prolong it. I want to be done too. Okay?
Sweet. Will we know our part one quiz grade today?
Part 1 quiz grade you will not know. And the reason is because the part 1 is all short answer. It will take me longer to grade those.
And so part B, will we know by the end of Thursday? Because like you inputted that grade.
First hour people, video, probably will. 7th hour, no. You won't know until the next day. Yeah. Okay, any other questions about that?
So like, when do we find out part 1? Like,
As soon as I can. I like to have it done. I was telling, I think, was it 7th hour? I was telling, I like to have grades done for you guys Friday before going for break. Was it 7th hour I told that to today?
yeah.
And the reason is in high school, the end of the semester, like grades are done like right then. I know have a little bit of a buffer coming back from break. Like teachers, we have like this extra week to finish grading everything, even though everything's due by the end of the semester. But I'm treating you guys like I would a high school course or a high school class because it's a high school course. So my grades for you guys, I would like to have them done by 4:00 Friday. Okay?
Never mind.
Okay. And if I haven't done before, then I'll let you know during our party. Okay? Any questions about the experimental design part? Okay. Let's see, what was next? Oh, characteristics of life. Okay, so characteristics of life. I did put up, 1st hour you didn't see it. I showed 7th hour this. I started putting up some videos. It's under final exam review. Do I need you guys to go out and watch everything? No. I would see what areas you're not sure about and then find the videos to go with it. I don't know. I took only the Amoeba Sisters because the response that I got from you guys was Bozeman is too much. You know, he goes into a lot of more detail. That's the Paul Anderson guy where you see his face. Yeah.
Like Amoeba Sisters.
It took me like an hour to get through it.
Yes.
Well,
Like 10 minutes long.
So I did give the Amoeba Sisters. If you want more detail or feel that you need more detail, I can give you the Paul Anderson link. I might even just put his general biology video link in there, and you can find the video that you need. Mine is no. I'm not going to do Hank Green because it really does go too fast. So my computer's having silliness, but you guys will see it in your class pages. At the beginning, it says at the end it says final exam review. I think this is going to stop on me. Okay, but characteristics of life was the next category. And so if something is living, there are certain traits that they all have. And one of the things that is brought up, if you do watch the Amoeba Sisters, is that there's no definite number. Okay, if you have to give reasons why something is alive, there's not a magic number. Like you have to have all 5 characteristics listed. It's always 5 or it's always 7. Because depending on who's talking, they divide it into different categories. And sometimes some organisms, you can't really tell. And some organisms, like viruses, they seem to have some of these characteristics, but not all of them. So there's always this debate. But in general, there are certain ones that are common. So anyone know something that all living things, animals, plants, fungi, bacteria, all living things have to do or have?
Cells or cell-like structures.
Okay, they're all based on cells. They all are made of cells, at least one cell, okay? Okay, and I'm going to write it singular, like bacteria or amoebas. You're talking single cellular. Or what do we call it if it's single cell? What prefix? Unicellular, okay? So they're composed of cell or a cell or cells. Now, yes, there's things inside those cells, but if we just took out like the nucleus of a cell, you wouldn't have something living. It would need other parts too. Okay, what else? So besides being composed of cells? Reproduction. There's different ways to reproduce. We'll look at that next semester. Asexual, sexual reproduction. But they need to be able to pass on their genetic information. What's their genetic information? DNA. Yeah. Okay, so and what's something else that they can do? And if you're not sure, you can throw them out there, Jacob. I have genetic information. That has to do with the reproduction, though, yeah.
This is going to go under cell, kind of, but a cell nucleus?
Nope.
Yeah.
If you have bacteria, they're living, they don't have a cell nucleus. Do you guys remember from last year what you call a cell that has no nucleus? No rhymes with what? Not go. It means before pro. A pro what? Prokaryotic. And if they have a cell, what are they called? Eukaryotic. Nice. Okay? So let's think about what we've been focusing on with Africa. What have we focused a lot on with these animals and even the plants here when we're doing our food webs? What do they all need? Yeah. Energy. Perfect. Okay? So all organisms use energy. Use energy to do daily functions, reproduction. So I'm just going to write down the word energy. These are general concepts. And like I said, sometimes you'll see some of these grouped together. Sometimes they'll break it down further. Okay, what else can they do? For instance, we do this when we go outside in the cold. What happens? We can regulate because can we sense that it's cold outside? Yeah. We can respond to our environment by doing what? Shivering. And we're going to talk homeostasis next semester, but we have to be able to respond to our environment. All living things can somehow respond to their environment, whether it's plants growing into the direction of the sun, whether it's the leaves and seed the wind and seed dispersal, okay? But there's a response, whether it's the sound of a predator approaching. There's responses and stimuli, and that's a characteristic of living things. Yeah.
Adaptation, like they need to be able to adapt.
Yep. So.
Or would that be part of?
Nope. And so this is one of those things. Sometimes things are grouped together. Sometimes they aren't. But I'm going to write that down. So I'm going to put they can respond to the environment, and as a result, they can. I'm not going to use the word adapt quite yet because we haven't clarified what it means to adapt. Or did we talk about adapt earlier this year? Adaptation, and about how you can't do an adaptation is not like you can't just want a longer neck. It's like populations over time are going to change the most favorable. Okay, so if we can understand that it's the favorable traits increasing in the number as an adaptation, that's fine. Otherwise, think of it as they change over time or changing over time. Yeah.
What are plant cells called, and what are
Plant cells.
And animal cells?
Animal cells, and it depends on what their specific job is.
Okay, because I thought for some reason I got it in my head that prokaryotic and eukaryotic were for animals and plants.
Nope.
Because I'm the dum-dum.
Plants and animals are eukaryotic because they both have a nucleus. Prokaryotes are bacteria and archaea. Mm-hmm. Archaea are ancient bacteria. So that is living things. Any questions about the characteristics of life? Okay, identifying common elements of life. This goes on that digestion lab, and we talked in first hour. I have to get my brain out of what I was taught, because you're right. I like using the way the Amoeba sisters used it because it's in order of abundance. All living things, though, have certain elements that are essential to life and common to life. That means that there's a great abundance of it. There's some elements like magnesium living things need, but maybe it's lower amounts that are needed. So what is one of the elements that, and I'm going to try to get 7th hour to answer these questions because 1st hour I covered kind of this with you today. So in 7th hour, think about our digestion activity and looking at cellulose and looking at the proteins, and we had to see what elements and count the number of elements that were there. What's one of the elements? Carbon. Okay. Madison. What is it? Yes. Nitrogen. Okay. What else does cellulose break into? What is it? Hydrogen. And there's one more that cellulose breaks down into. Michael? Oxygen. The last two being phosphorus and sulfur. Amoeba Sisters calls it chomps, which is awesome because carbon, hydrogen, and oxygen are the trifecta that everything has. When I say everything, all classes of biomolecules. All classes of biomolecules have carbons, hydrogens, and oxygens. So what are those 4 classes of biomolecules? Yeah.
You got your carbs.
Okay, so carbs. What else?
Your protein, nucleic acid. and then it's
Lipids.
yeah. But we both know that it's schnapps, right, Mrs.?
Yeah, I learned it as I was telling them. I learned it as schnapps. I don't know why. Chomps does make more sense, but I'll still call it schnapps sometimes by mistake. Not by mistake. It's just what I was.
Can we
Oh, it's
educate people as to what schnapps is?
No, Christian. Okay, so I'm going to cover this on Tuesday more in detail, the biomolecules. I know I have some people doing text corrections. But you'll have carbons, hydrogens, and oxygens in all of them. Now, nitrogen. What kind of molecule was nitrogen found in when we did the digestion lab? I'm going to have someone else. Think about digestion. Was nitrogen in cellulose? Was nitrogen in the cellulose when it broke down? No, it wasn't. Well, okay, so cellulose. Was cellulose a carb, a protein, a nucleic acid, or a lipid? Cellulose, a carbohydrate, which is a sugar, a protein, a nucleic acid, or a lipid. Jacob?
Nucleic acid.
Carbohydrates. So really quickly, in case you don't come next week, little quick carbohydrates. Carbohydrates are sugars.
Do plants have
Okay.
lipids?
We have simple sugars, yep, and we have complex. We'll talk about those next week. But a key identifier is this suffix. You can tell that you're dealing with a carbohydrate without ever seeing the element or the molecule before by seeing the ose. Cellulose, glucose, fructose, sucrose. Okay? It's a common identifier of carbohydrates. So ose means sugar or carbohydrate. Now, nitrogen, though, was not in cellulose. What other macromolecule were we focusing on in the digestion lab? What did you have to break down and put together? When you cut those squares apart and broke some bonds? Proteins, okay? So here we go, proteins. Now there's a couple of proteins that have there's a couple of amino acids, but not that are. It's mostly this. Nitrogen is in every single amino acid, the building blocks of proteins. Okay, nitrogen is characteristic. If you see a nut in there, you're probably dealing with a protein. Now there are a couple of amino acids that have sulfur in them, but don't stress about that. Okay, the key here is if you see an N in the molecule, chances are you're dealing with a protein. If you aren't sure of anything else and you just had to go with that as your only knowledge, go with protein. Okay? The other ones, and CHO is also lipids. Okay?
Wait, is carbs just?
Yeah. The last group, nucleic acids, will have all of those. All of the chunks. Nucleic acids, we really haven't talked much this semester about. They are, nucleic acids are DNA. Okay, that's one, and RNA. Yes. Next semester, we'll talk nucleic acids in more detail, like the structure and whatnot.
Okay, so our guanine, cytosine, and all of that good stuff, are those nucleic acids or like?
Those are nucleotides, which are the building blocks of nucleic acids. So nucleic acid is the biomolecule. That gets confusing because what are the building blocks of proteins? So think of my t-shirt and think of
Amino acid.
amino acid. So amino acid is the building block of a polymer, but nucleic acid is a polymer that's made of something else too. So sometimes the word acid kind of trips us up. Okay, questions on that? Yeah.
Not specifically on this, but will we have to memorize the monomers of these
Eventually, you should know.
for the final project?
What is the monomer of carbohydrates? What's the simplest sugar here? Monosaccharide, one. And what is the one that we commonly talked about, that we talked about a lot, like during photosynthesis? It's one, like when in doubt, use this as your monosaccharide, glucose. Okay? They're monosaccharide simple sugars. Amino acids are the building blocks of proteins. Fatty acids are the building blocks of lipids.
Which is like, was glycerol the name for all of
Glycerol is the head of like the head part of it. Okay, any questions? Okay, so we only have a few minutes left. We're gonna kind of go through this kind of quickly. This first part, pyramids have to do with a couple of things. They can deal with it. We've been talking about energy. They can also be pyramid of numbers. But in either case, what happens to the shape as you go up it? Smaller. So whatever it's measuring also gets smaller. So energy, as you go up it, what happens to the amount of energy available at each level? What? Less. Okay? So what uses the sun's energy? All energy starts with the sun, right? So which level actually gets the most energy here, do you think? Michael? Producers. So they generally, they utilize all that they can. So we usually think of that as 100%. That's our starting point. And then every time you go up it, every time something consumes, so our primary consumers consume the plants. Our carnivores, which are secondary consumers, consume the herbivores. We lose energy. And what rule do we have for that? What percentage? 10% rule. Only, even though this is definitely bigger in the pyramid than 10% of the bottom row, every time you go up, okay, 90% is lost. It's either used to maintain the organism or lost as heat, and only 10% is able to be utilized by the next level up. Yeah.
How does one measure the amount of energy in a producer?
Same as others. Kilocalories are the general way of measuring energy in anything living. And so when you're looking at kilocalories, what you generally do for plants to see how much energy is you look at the mass, because that mass is called biomass, like you dry it out and whatnot, and you can determine the amount of energy that way because of the amount of glucose, which is that stored energy, the amount that's been converted over. Does that help a little bit? If you ever go on to AP biology, you will look at, primary consumers. Not primary consumers. You'll look at, I'm having mind farts. Primary consumer energy, but you also look at producers and calculating energy using biomass.
What's the difference between actual bio and just AP bio?
AP is like college level. So this is like an introductory high school
So
biology.
we take all of our high school classes and then we take AP bio, AP like.
So yeah, so what's happening for you guys? And this is kind of a side note. Did you guys want to? We have about 2 minutes. Do you guys want to? Oh, really quickly. Here's a 10% rule in more detail. Okay, so you can see it here. Decomposers. They kind of get a little bit from all over the place. Okay, they're also not depending on where they fall. Generally, when we do these pyramids, we don't always include them because they're just kind of their own creature. They are part of it, but you can see that they're kind of off here because they take anything that's dead. We'll talk more about them with carbon and nitrogen cycle. Food webs, make sure that you include, and I'll talk about classes in a minute. Make sure the biggest trip up here, and even though it may seem obvious, the arrow is not what eats what. What does the arrow represent? Zoe? Energy transfer. The energy is moving from what to what? The energy is moving from this tree to this deer, from this deer to this leopard. Okay, think of it in terms of that. Yeah.
Well, it has a higher population density, I guess you would say, an apex predator or a decomposer, like a vulture as compared to a lion.
Decomposer. Yeah, decomposers. Okay. I am going to stop there because then it goes into populations, which we're just hitting in the first hour we'll hit. And it's about 3:44. So what I'm going to do is I'm going to stop this, but if you have, I can talk to you about your courses for a second if you want that answer.
Because there's, well, like,